Welcome friends to another interesting video. Today we are going to talk about a cell organelle. So previously we have talked about mitochondria and today we are going to talk about a cell organelle. The unique feature of this is it is going to be present both in prokaryote as well as in eukaryote. I believe people are aware what a prokaryote means. These are a primitive cells with an developing nucleus, whereas eukaryotes are the one with an advanced nucleus, right? So here it's going to be a bacteria, and eukaryote it is going to be protista, that is amoeba, fungi, plant, animalia. So most of the organisms do come under eukaryotic domain. So this organelle, as I said, it is present both in prokaryote as well as in eukaryote. Okay, hope you would have guessed that the organelle is going to be ribosome, right? So let's discuss the various aspects of ribosome in this particular video. Okay. If you consider this as a cell and the center part, obviously it's going to be the nucleus, and you call this region as cytoplasm. This cytoplasm is known to contain many organelle, which we commonly call it as cytoplasmic organelle, like as I said, mitochondria, chloroplast, endoplasmic reticulum, etc., etc. And one such organelle is going to be ribosome. As I said, the unique feature is that it is going to be present both in prokaryote as well as in eukaryote, but there are going to be some minute differences in ribosome of the prokaryotic version as well as in eukaryotic version. So let's discuss all those things in this video. Friends, you have to know one thing: wherever a DNA is present, for the DNA to completely execute its function, ribosomes are required. So when you see a DNA, you should be able to see a ribosome, right? So obviously DNA is seen inside the nucleus and ribosomes are seen outside the nucleus, that is in the cytoplasm. Okay. So maybe when in higher concepts you will get to know the exact reasons behind this concept what I am saying here, right? So with this, let's get into the ribosome part. Friends, this ribosome part, okay, basically it has got no membrane. Okay, the ribosome has got no membrane. Usually in cell biology, we are supposed to take an account of whether an organelle has got a membrane. Yes or no? No, fine. In case if it is yes, then whether it is going to be a single membrane or a double membrane. Say for example, mitochondria, chloroplast, everything has got a double membrane. So, that view on organelle should always be there. So, with that, the ribosomes are the one without any membrane. There are no membrane. So, then what is it made up of? It is made up of two components. One is going to be an RNA called as ribosomal RNA, which you call it as an rRNA, and it is made up of, other one is made up of protein. Okay, rRNA and protein. And friends, as I said, this is present in both prokaryote as well as in eukaryote. So, there is always a comparative analysis should be done. And the prokaryotic, the ribosomes are, the rRNA is going to be 50% and protein is going to be the 50%, whereas in eukaryotic version is going to be the 60% and this is going to be the 40%. So this is one major difference that you see between the prokaryotic one and an eukaryotic one. Okay, right. Second one is what is the exact structure? If you look into the, not exact structure, a rough structure, you should be able to observe that this ribosome is made up of a structure something like this, which is called as a smaller subunit, and you call this as a larger subunit. Okay, a smaller one and a larger one. And usually only when there is a need, both the subunits will be joined together, otherwise both will be separate. And the one ion which is required for the association of both the subunit, it is going to be the magnesium ion, which is Mg2+, right? So this is one important point. As I said, we should always have a comparative analysis, right, between the ribosome version of prokaryote and ribosome version of eukaryote. So if you see, You can say smaller, larger, can also include total one. The prokaryotic version has got 30S, 50S in larger. S stands for Swedberg unit, which is nothing but a sedimentation coefficient. Okay, something related to biophysics. So we don't have much time to describe about the sedimentation coefficient at all. So just remember that the S stands for Swedberg unit, which is nothing but the rate of sedimentation or sedimentation coefficient. Okay. And friends, the total actually should be 80 according to mathematics, but it should be 70S. So the smaller is going to be 30S, the larger is going to be 50S, and total is going to be 70S. This is going to be in case of prokaryote. Whereas in case of eukaryote, this is going to be 40S, this is going to be 60S, and total is not 100. People always say your mathematics is not applied here. Okay, it should be 80S. Okay, so this is the smaller, larger unit versions of both prokaryote as well as in eukaryote, right? So these are all going to be the structural aspects. As I said, both subunits, I repeat again, they are not associated always, they remain separate, and whenever there is a need, both subunits will associate with the help of magnesium ion. Hope you get this point. Now, coming to the function, what does the ribosome do? Coming to the function of ribosome, friends, I have informed in this discussion that wherever you see DNA, okay, you will be able to see ribosome. So DNA without ribosome or ribosome without DNA is completely useless, right? So then what is the exact the function of ribosome? Friends, you can observe that this nucleus is going to have DNA. So I'll write it here. This is going to have a DNA. Now from this DNA, okay, through a process called as transcription, obviously these are all higher biology concepts, but just memorize in case the viewers are of a bit lower classes. Okay, so understand that from DNA the information is carried in RNA, which is called as messenger RNA or mRNA, by a process called as transcription, right? Where this happens, okay, this is called as transcription. And where does this happen? This happens inside the nucleus. So mRNA is ideally formed inside the nucleus. Okay, so this is there inside the nucleus. So friends, what happens is the mRNA, once it gets out of the nucleus, and you see the ribosome, the smaller one, larger one, both will be separate, right? If this is going to be the mRNA, the mRNA associates itself with ribosome like this. You can see structure like this, right? So this mRNA will get translated. As I said, these are all bit higher concepts. So people about 12th standard, will be able to understand these concepts, right? So you get a structure here, which is protein. So you call this process as protein synthesis. So obviously ribosomes can be considered as a factory of protein. It is the place or the organelle where the protein synthesis do happen. So for which the information is obtained from DNA, which is there inside the nucleus, through an RNA, which is called as messenger RNA. Hope you get this point. So this is going to be the sole function of ribosome, protein synthesis. So this is going to happen when ribosome gets the information. I repeat this again, and that information is contained in the RNA, and that RNA is obtained from DNA through a process called as transcription, right? People will be dealing this, okay, in detail in 12th standard, right? So this is what is called as protein synthesis, or I can also call this as a translation. Okay, can also call this as a translation. So this is the function of ribosome. And friends, ribosome can be seen isolated in the cytoplasm, which we normally call it as a free ribosome, or it can also be associated with endoplasmic reticulum, where the endoplasmic reticulum will be referred as rough endoplasmic reticulum. Okay, wherever it is, mostly the function is going to be the protein synthesis. And friends, in one case, what happens? The ribosome, say assume there is going to be mRNA. Usually what happens in one mRNA, only one ribosome will be present, but in some cases, one mRNA will have more than one ribosome. So you call this as a polysome. Okay, we call this as a polysome. It is, some books says that it is seen in prokaryote, but in general it is seen in prokaryote as well as in some eukaryote, right? So hope you get a clear picture regarding ribosome. So in this video we have talked about the ribosome, its nature, a little bit of its structure, right, and its function. Hope you get this point. In case of any doubt, so please do mention it in the comment section, or please do mail us. We'll be there to help you, right? So we'll meet, okay, in another video with an interesting topic. Thanks for joining.